environmental niches, Entelis in northern India, Hypoleucus and Priam in South India, and then in the Western Ghats you have Jonai, that which is Nilgiri langur. So environmental data, morphological data, and genetic data all support splitting Hanuman langur into at least 3 different species, one in northern India and 2 in South India. Right. Now, what about the Himalayan langur? So this subsequently we extended this work to include Himalayan langurs. So these are langurs confined to the Himalayas. So we sampled from Kashmir to Sikkim. And once again, you have a distinct clade of Himalayan langurs, a distinct cluster, genetic cluster of Himalayan langurs that are distinct and different from the langurs in the plains. The langurs in the plains, the one called Entelis found in northern plains. So these two are completely distinct, and we used the same approach, looked at morphological character and ecological character, and found the same result. So here's the overall synthesis. What we used to call as Hanuman langur, and we thought was a single species distributed from the Himalayas all the way to Sri Lanka, actually consists of 4 different species, right? And we have ecological, morphological, and genetic evidence to support this. In the Himalayas, we have Semnopithecus cetaceus. In northern plains, all the way through central India and parts of Orissa, we have Semnopithecus entelis. In South India, we have Semnopithecus hypoleucus, and further south, we have Semnopithecus priam. So, thank you, everyone.
Thank you so much, sir, for fantastically, summarizing the outcomes of the research that you had taken on the taxonomy of different Hanuman langurs and how it is relevant and related to different places in India from north to south. And this was fantastic insights. And this is very much important aspect, from climate change viewpoint also, that how this taxonomy or this distribution is being affected with changing climate. So that could be another interesting area that we could think of. Thank you so much,
Thank
sir.
you.
Now we are moving to next. This is turning carbon into livelihoods. Algae-powered biomanufacturing for green jobs and reduced synthetic nitrogen dependency. The speaker is Dr. Adhi Viswanathan from Pinnacle Biosciences. So this is over to you, sir. Kindly finish it in 5 minutes.
Yeah,
thank you so much.
Start sharing the result. Good evening, respected advisor, Biomanufacturing Directorate, Secretary DBT, and distinguished guests, and fellow researchers and industrial leaders. Today, I would like to share how algae-based carbon capture and utilization can address the 3 major critical issues today India is facing. Traditionally facing, like first one is industrial carbon emission. Second thing is our the chemical fertilizer crisis that we are that is happening today. Third is employment generation. Traditionally, these 3 challenges can be addressed independently. We believe that algae biomanufacturing offers an opportunity to solve them together. So that is what today I wanted to present here. So the chemical fertilizer crisis, the global consumption of the chemical fertilizer today has been accounted to be 205 million metric tons, whereas 50% of them is nitrogen fertilizer. And coming to the average nitrogen use efficacy, the global standard, if you are seeing, it is only 55%. And approximately 50% of the nitrogen is being lost. It is not available to the plant. So coming to the consequences of these chemical fertilizers that we is happening today is, see, like 90% of our chemical fertilizer is being imported. In India is being imported. We import approximately 15 million tons of chemical fertilizers annually. So which results in rising of import bills, high fertilizer subsidies, soil degradation, water pollution, nitrous oxide emissions and supply chain vulnerability as like the war times, which we are facing. Coming to the economic impact, India spends approximately 1.7 to 1.9 lakh crore annually on fertilizer subsidies. So we are looking for an alternate way where India's fertilizer challenge is no longer an agriculture issue. It is simultaneously on a food security, economic security, and climate change. So we propose algae-based solutions for sustainable agriculture, where these chemical fertilizers can be gradually replaced by these algae-based products. Why algae? See, these algae are primitive plants which can grow in areas where agriculture cannot happen. And any kind of water, like saline water, wastewaters, can be used to cultivate algae. And they are like 10x more faster when compared to that of land plants and forests in their growth. And it, as these algae are single cell protein, it is a whole nutrition where it has good protein, nutrient like minerals and sugars, so that when it is being used as fertilizer, the absorption of the nutrients to the plant is the availability is very high. And when it is used in soil, it enhances soil microbial activity. And also now, to have a net zero agriculture, these algae based fertilizers, mainly these blue-green algae, which has this heterosis, which absorbs atmospheric nitrogen, can be used to have carbon negative agriculture and so on. And apart from nutrition, these are all also acting as a very good biostimulants. So apart from nutrition, it also gives a very good biostimulant property to the plant. So it can be a very good replacement for synthetic fertilizers, and that as the crisis which we are having today. The objective of our project is not to completely replace the synthetic fertilizer, but to produce more food with less imports. Coming to the capital to biomanufacturing, so we'll have a small max. So if India substitutes 10% of its imported fertilizer, which will be approximately 1.5 million tons of chemical fertilizer into algae-derived input, the sources of our the algae cultivation is our carbon dioxide, and the sources of carbon dioxide may be the thermal power plants, distilleries, cement plants, fermentation industries, or biogas plants. So to grow 1 kg of algae, approximately 1.3 to 1.8 kg of carbon dioxide is required. And 1 kg of algae will be approximately having 30 to 40% of protein. And when it is converted into nitrogen, it will be around 4 to 6% of nitrogen. So which is very similar to that of nano urea, which has 4 to 6% of nitrogen. And again, the highlight here is not only the nitrogen, because today the nitrogen availability, the absorption of nitrogen is a big question. So as algae are single cell protein, apart from nitrogen, this also has good amount of minerals and sugars, which will make this nitrogen absorbed by the plant. And here is a small max, like when we are substituting 1.5 million ton of fertilizer into an algae-based product, we'll be growing 1.5 million tons of algae, which will be internally sequestering 2.7 million tons of carbon dioxide. And for every ton of CO2, we can give one microbiologist or biotechnologist, a job can be provided. So it means that approximately 2.7 million jobs can be provided just by sequestering 1.5 million tons of, by producing 1.5 million tons of algae. And the oxygen generator is huge. It will be like 1.8 million tons of oxygen is being synthesis. So these are all the benefits when we are substituting algae as a carbon capture. So algae transformation carbon emissions for a liability into a biological feedstock. So we provide the industries and solution which generates a revenue to the industry, unlike other chemicals like amine technologies and all those things. So this is the process which we are doing for our algae production. So we have introduced two unique proprietary system in this industry, which is a carbon dioxide infusion system. As CO2 is a poorly soluble gas, we have designed a system where CO2 is efficiently dissolved in water, and that water is used for algae cultivation. And we have a unique methodological high-raised algae culture system, which is like a competitor, or which will be much more better than the raceway tanks and the photobioreactor, which is being conventionally used in algae cultivation system. And this is our system which we are proposing. So this makes algae cultivation very feasible and economical in large scale CO2 sequestration and algae cultivation. And we already have a product which is made from microalgae digested, protein digested into amino acid, and which is already in the market. So we are just going to like copy paste this in a large scale. So that is what we are trying to do, and we are with these industries we are discussing to make this algae CCS happen and to give a lot of green jobs for our fellow Indians. So for concluding statement, the future of sustainable agriculture will not be defined by how much nitrogen we apply, but by how efficiently we use it. And the future of climate action will not be defined by how much carbon we intake, but how the carbon is being valued, how the value chain of the carbon is happening. That is what we are working into. Thank you, Jai Hind. And thanks for providing this opportunity on this Global Environmental Day. Thank you.
Thank you very much, sir. This is fantastic approach to convert or make available the sustainable carbon for the agriculture purposes, or you can say repurposing of the waste carbon for benefit of the agriculture and reducing import dependency as well as towards Atmanirbhar Bharat while fighting with the climate change too. Thank you so much for being with us. So now we are moving to serial number 3. This is closing the carbon loop, transforming CO2 from biomethanation into value. Here the speaker is Dr. Manju D. Tanwar from Organic Recycling Systems Limited. So it's over to you, madam. Kindly finish it in 5 minutes. Thank you so much.
Thank you. Thank you so much. Can you see the screen, sir?
You can share. But we can't see right now. We are unable to see. Yes, I think go ahead. Yes. Now we can see, just make it full screen mode and go ahead. Yes, please go ahead.
Yeah.
Perfect.
Yeah. Good afternoon, everyone. Today, I would like to thanks to all because DBT Bayrek, I'm participating this event today. So I'm really thankful to carbon loop, and it's connected to the BioCCUS project also, which has been awarded from DBT Bayrek. So under this, actually, first, I would like to introduce a little bit about my organization. Organic Recycling Systems Limited has established in strong presence in bioenergy sector. So particularly in biomethanation process, where organic waste and agriculture waste is converted into valuable resources, specifically bio-CVC and biogas through electricity. So under this project, we have proposed, because biogas contains carbon dioxide and methane, both almost in similar amount. So CVC is utilized as a replacing fossil fuels, but carbon dioxide unutilized. Still, people are searching about the better technology to utilize this CO2, but it's still not utilizing properly. They are trying various technology for converting this CO2 into value-added product. Under this project, we have proposed two approaches. One is biological approach, that is biological fixation through algae, and the second one is photocatalytic approaches, where CO2 is converted into mixed alcohol through photocatalysis. So this is the project. I'm not taking much time for this. So basically, the theme of this project is we are not focusing on the CPG, we are focusing on CO2 only. So after separation, CO2 can be utilized and diverted to two reactor streams. One is photocatalytic reactor, which is converted through photocatalysis and visible light, presence of the visible light. And the other approach is biological fixation, because algae cannot utilize entire CO2, because if we will consider 100 ton of organic fractions of MSW plant, so it's around 8 to 9%, 8 to 9% CO2 generated every day per day. So it cannot be utilized entirely through algae. So that's why we have used photocatalytic approach also here. So maximum we can utilize this CO2 into converting into value added product through photocatalytic way. So here I have given few benefits what happen if we utilize this. So instead of waste, CO2 become a secondary feedstock of value addition of the biomethanation plants. So because of that reason, up to 20 to 30% increase overall carbon utilization efficiency increases, economical benefit and environmental benefit also we through this particular technology. So this is actually my project. I only take 5 minutes only. Thank you so much, sir. Thank you. This is my project. Thank you.
Thank you so much, madam, for looping the carbon emissions that is happening out of the bioenergy process and presenting this bioenergy integrated carbon capture, utilization, and storage thing. This is very much important for looping this carbon and making the biofuel production processes or bioenergy processes more cleaner as compared to today. So thank you so much. Now, actually, there is some request. Somebody is from outside, so we are taking number 10. This is dry times at forest ages, seedling traits and response to drought. Dr. Meghna Krishnadas from National Center for Biological Sciences, Bangalore, would be presenting here. So it's over to you, Dr. Meghna Krishnadas. Can you hear us? So I think.
Dr. Meghna, are you there?
There is some connectivity issue, so we can continue with the number 4. This is, can the oldest bacteria help make biomanufacturing sustainable on Earth and space? So Dr. Sri Srivastava from International Center for Genetic Engineering and Biotechnology, New Delhi, is the speaker here. So it's over to you, Dr. Sri. Thank you so much. I think we request you to finish it in 5 minutes only.
Yeah. Can you see my screen?
Please go ahead. Perfect.
Yeah. Good afternoon, everyone. And first and foremost, thank you to Dr. Sangeeta Kasturi and Dr. Balaji for giving opportunity to present our research, which is on can we repurpose the oldest bacteria to help biomanufacturing sustainable on Earth and space. So now Dr. Adhi Visvanathan has already set up the problem for me, and we are also trying to address the same problem that our country and the world is currently facing, the carbon and the nitrogen challenge in terms of sustainability. We are emitting too much of carbon dioxide, leading to global warming. India is the third largest emitter of CO2. And on the nitrogen challenge, actually, we are also a third largest importer of urea in the world, which is provided to the farmers so that they can grow their crops to feed the population of humans as well as cattle. Interestingly, a lot of nitrogen is excreted as urea and urine. And also, very interestingly, actually, instead of being an importer, we can in fact become a urea exporter if we can recycle the urea from urine. When we talk about bioprocesses, the same is there, that if we can recycle the carbon and nitrogen, we can improve the sustainability of our bioprocesses as well. So, and not only on Earth, even on ISS, the International Space Station as well, there is an efficient system of recycling of water, but there is still no efficient method for recycling carbon and nitrogen. And this is one of the major challenges in designing long-term human stays for space. So with this background, I want to briefly introduce cyanobacteria, which are the oldest and fastest growing phototrophs. These were actually the first photosynthetic organism with fossils dating more than 3 billion years old. And at that time, Earth was very rich in CO2, Earth's atmosphere, and these cells grew under those conditions. The long evolution has also helped them to become really, really robust and also very efficient. So unicellular strains are the fastest growing phototrophs. There are also many marine strains which we are focusing, and they would not need fresh water. And being photosynthetic, they can utilize carbon dioxide, and we can also make them utilize urea to produce oxygen and a nitrogen and antioxidant rich biomass, which can be used as a fertilizer, food, and feed. The challenges on using cyanobacteria for carbon capture and recycling with the real industrial kind of waste is that the flue gases have very high CO2 concentration as well as sulfur dioxide and nitrogen dioxide, which is basically toxic to these cells. And the same is the problem with urea in urine, which is at very high concentrations. So this would actually leads to bleaching of cell as shown here. So what we have done is we have used a combination of adaptive lab evolution as well as process optimization to ensure that these cells can tolerate high CO2 and high urea concentrations. So here is where we are showing one setup where we have utilized an indigenous marine cyanobacterium to capture carbon from a simulated flue gas. This cyanobacterium was adapted so that it can now tolerate high CO2 concentrations. And we have used a very high CO2 concentration of 18% in the flue gas, simulated flue gas. And with a single pass, we were able to capture over 96% of the CO2 using this indigenous marine cyanobacteria. We are also very fortunate to be a part of the Axiom 4 mission last year, which was part of the DBT's BioE3 policy, where our experiment cyanobacteria on ISS was conducted aboard ISS, and ISRO and ESA were also involved in executing this experiment aboard ISS, where we compared the growth of two cyanobacterial strains on urea or nitrate under microgravity conditions. We included an Indian isolate of spirulina and a very fast-growing Senecococcus strain in our study for their obvious beneficial properties. Because manuscript is in preparation for submission, I will not go into the details of the results of this, but give you a snapshot of what we have done. This is how our hardware looked, which contained the two different types of cells. And this is astronaut Shubhanshu Shukla conducting our experiment aboard the ISS. Not only this experiment, we have continued our work in this area, and now we are able to grow cells under much greater urea concentrations, approximately 10 times higher than what we had done before. So with that, I'd like to thank the Department of Biotechnology for the support that it has provided, ICGB Director, Faculty and Administration for the support, NFMC of Bharatidasan University for providing native cyanobacterial cultures, and also a lot of my students who have worked in this area and have made this successful, as well as my collaborators. So our overall approach can be summarized in this slide. We are aiming to usher in an era of photosynthetic biomanufacturing where CO2 and urine are not waste, but a resource. Thank you.
For this integrated approach, so you have connected biomanufacturing with all three, Jal, Thal, or Nav. It's like this. So this is very much integrated. Thank you very much for sharing the insights. So now we are moving to next. This is serial number 5. So here speaker is Dr. Krishna Ray from West Bengal State University. This is restoring biodiversity in Sundarbans mangrove niches, success with indigenous plant growth promoting rhizobacteria. So it's over to you, ma'am. We will request you to kindly finish 5 minutes. I think you can share entire screen and then just
Yes,
move to your
I can
slides.
do that.
Yes.
I cannot find it. Which one is this?
No, I think. No. You just open the presentation
Yes, I have opened it. Then
and then go to the share button. Share is there, na?
share screen.
In
I'm
bottom
going
panel.
there. I'm going there.
Just go to
Then
share.
it's showing your screen.
Yes. Entire screen.
Yes, now it has come. Okay.
Yes.
Can
Perfect.
see this?
Go ahead.
Okay.
Yes, we can
So,
see. Perfect.
thank you, DBT and BIREC team, for giving me this opportunity to talk here. This is about restoring biodiversity in Sundarbans mangrove niches, success with indigenous plant growth promoting rhizobacteria. Under the DBT support, since 2014, we have been working on a mangrove biorestoration project in Indian Sundarbans. And under this project, we have so far, we have restored around 65 hectares non-protected degraded mangrove niches, and we have converted these niches into which were earlier degraded from where the mangrove species were already almost lost. On those sites, we have converted them to multi-species self-sustaining dense forest ecosystem which are resembling just like mini Sundarbans. And each year thousands and thousands seedlings, they are coming there, being recruited naturally, and they're making the sites more denser each season, and thus we can see that the ecosystem got repaired which were earlier damaged. So not only that, under this project we were successful in reintroducing, rather we can say reintroducing, several rare and threatened species which are already lost from the shorelines of these non-protected shorelines of Indian Sundarbans, like Heritaria fomes. This is endangered species. This Sundari, Sundarban is was the name and after the name of this species, but now we cannot find them actually in the western part of Sundarbans. From the shorelines, it's totally lost. But we have been successful in creating germplasms of this germplasm source of this Sundari now in several of our sites. Not only Sundari, Phoenix paludosca, this is also a near threatened line, brown layer tursa, Insia ajuga, these are all under threatened category, and some locally rare species also, Nipa fruticans, Meropangulata, Xylocarpus, Lumnizeda racemosa, and many others. We have now our sites have become the source of germplasm for these individual species. But what we have seen that all these species, they're not showing uniform growth in all sites of restoration. So some sites are very much challenging and where they're just surviving and their growth is becoming stunted. So what we then for to improve the growth, what we have added in our restoration experiments, we have added one component by application of indigenous plant growth promoting rhizobacteria. Over the last decades, with DBT support, we have been able to isolate almost 400 bacterial isolates from the rhizospheres of Sundarban mangrove niches, and we have screened them in the lab under standard experiments, their plant growth promoting activities. And from that, we have started screening these PGP rhizobacteria, and we have started applying them to the degraded mangrove sites. So here today, I am just going to tell you about one experiment where we have taken two consortium. One consortium was entirely made up of actinobacterial isolates. We actually wanted to see whether these actinobacteria solely it is able to improve the growth of the mangrove or not. So this, and then we made another consortium. This is consisting of non-actinobacteria, mainly proteobacteria, gamma proteobacteria, this and firmicutes. They were included under these, and we have screened all of them under laboratory conditions. Many of them are good nitrogen producer, nitrogen fixers. We have tested them by ARA assay, all these things. So what happened? We actually made large-scale cultures, and we have immobilized the cultures in biochar using the sodium alginate, and we made the beads. We took them to the our sites and we applied them. This is how we applied them on our sites, and you can see even after 28 days of so we could trace those biochar beads which are holding the bacteria, because there is a chance of washing out in Sundarbans. If you apply the bacterial culture, there is always a chance of washing out. So for that, we always immobilize this biochar, this bacteria on these biochar beads. This was totally these protocols were invented by our group of students, and they have also invented one indigenous machine for putting it in large cultures, putting it in large amount, because we need a large number of these biochar beads, bacteria immobilized beads for application. So I'm telling you about one experiment. This started in only May 2024, and this is a December 24 picture. You can say from here we have started. So here at this time they are same. They're the applied bacteria applied sites and without bacteria applied sites, they're almost same. But now this is a very recent picture on 12th April. You can see how the bacteria non-bacterial applied sites, these control sites, their growth is much less as compared to the sites where we have applied the actinobacterial consortium and also the non-actinobacterial consortium. You can see this is on the experiment was done on Phoenix paludosum, another near threatened line. These are totally on-site experiments with 75 plants each under each category, and you can see that this actinobacterial application after actinobacterial application, plants are palms are basically a very slow growing species, but here you can see how they are moving to the forming the pinnate leaves, and this is also almost after 2 years we could achieve this thing. And this is the above ground scenario. You can see always the actinobacterial application is doing better performance, significantly better than the other control and non-actinobacterial things. But what is happening at the below ground? What is the bacterial communities changes occurring? So for that we have done next generation sequencing, and here
sorry to intervene, ma'am. We request you to kindly yes,
you can see
we
have seen a very interesting observation where we have applied actinobacteria. There the indigenous actinobacterial population got decreased, but the Firmicutes population, it got increased. And where non-actinobacterial population we have added, there we have seen that actinobacterial population got increased. So this is how we think this is the mechanism how they are going to improve the bacterial this plant growth, and we are doing further experiments on this, and we are going to metatranscriptomics now to exactly showing what are the PGPR genes and from which bacteria they are going to contribute there as a for improvement of these plants. And we have particularly found that one actinobacteria is having higher population in non-actinobacterial sites, and another Firmicutes is having higher population in the non-actinobacterial in the actinobacterial sites. So this is a very interesting observation. We are still continuing with these experiments. So these are our field staff, which was supported by DBT and my students and the collaborators. I thank you for DBT for supporting us since long for doing this work. Thank you.
Thank you so much, ma'am, for showcasing this fantastic work. And Sundarban is one of the biggest carbon sink of the world, and really you are contributing in those harsh conditions. This is very much important activities. How it can be taken up, we understand it very much. It is very important initiative. Activities being taken up by you under DBT supported initiative. Thanks a lot for sharing these insights. Now we are at serial number 6. This is biochemocatalytic transformation of biorenewable resources into green transportation and sustainable aviation fuels, from Professor Azaz Ahmed, Indian Institute of Technology, Indian School of Mines, Dhanbad, IIT ISM Dhanbad. So it's over to you, sir. We will request you to kindly finish it in 5 minutes.
Yeah. Thank you, sir. Thank you so much. Am I audible?
Perfectly audible. Please go ahead. We can see your slides.
Yeah, thank you, sir. So here I will be discussing briefly about the biochemocatalytic transformation. But before I start, I would like to thank the DBT and the committee members for inviting me here. Most of the problems that I prepared has already been discussed, so I will skip them. What are the major problems? One is definitely the solid waste. The other is the greenhouse gas emissions. And as Dr. Joshi rightly highlighted, there is one more thing that we are missing is the methane emission, which contributes roughly to 16% of the total greenhouse gas emission. So, in order to um, I can say, in order to tackle these problems, we have found another way that instead of thinking the agro residue or the kitchen waste or the other things as a waste material, can we consider it as a resource material? And if you see here, almost everything that we can make from a fossil fuel, like chemicals, energy, fertilizers, or anything that we're making at present, can be prepared from the agro residue as well as the kitchen waste, etc. In particular, if I talk about uh, we have solid fuel we can make bio oil, syngas, power, biochar, or we can make high value chemicals that includes the aviation fuel, green gasoline. And in this regard, we got a project from the DBT that was particularly on the integrating the biological and the chemical aspect of engineering together to make the aviation fuel. And the reason is obvious: approximately 10 kg of CO2 is emitted per second when a commercial aeroplane flies. So herein, IIT Delhi, IIT ISM Dhanbad, and IIT Dehradun came together to develop a process that basically synthesizes the biomolecules by the fermentation route, and then we apply the butyric acid. We use a chemical catalytic route to convert that into the aviation fuel. Now, after converting that into aviation fuel, we also test it whether it is stable or not. And what we found that here, this up to 93% conversion can be achieved when we are trying to convert this biomolecule into a suitable aviation fuel, that is called butyl butyrate. And this butyl butyrate can be blended up to 50% without changing anything in the existing aviation engine. So that actually helps to reduce the CO2 emissions or the greenhouse gas emissions 2% contribution of aviation industry to 1% straight away if we use the biomass molecule derived fuels. Herein, like, we already have several patents and publication coming out from it. But motivated from this, what we did, we thought that this butyl butyrate is made from butyric acid and butanol. So can we make similarly for the gasoline and diesel? So we took up a project where we make the alcohol and levulinic acid together. And what we do, we develop a product called ethyl levulinate, which is a replacement of the gasoline. And this molecule, the ethyl levulinate, actually replaces the MTBE and polyaromatics. And for that, we also thought that there is a lot of requirement for chemical catalysis. So is there any way that we can find that what is the most suitable descriptor that actually helps to decide a catalyst without doing a lot of experiment? And we found that deprotonation energy, that means the energy required to take out one hydrogen from the catalyst, is responsible for the activity. And we tested it here for a lot of alcohols like ethanol, propanol, butanol, and we had two catalysts. One was the phosphotungstic acid, phospholinic acid. And what we found that the difference between the phosphotungstic acid and phospholinic acid was exactly 18.6 kilojoule, which is equal to the difference in their deprotonation energy. So this is activation energy, this is deprotonation energy. This indicates that if I know the deprotonation energy of a catalyst, I can easily find out whether this catalyst will be active or not. The second thing, when I'm changing it to ethanol, it becomes green gasoline. If I'm changing to butanol, it becomes green diesel. If I change some other reactant, it becomes aviation fuel. So there also we can find out whether this catalyst will be active or not. And for that, what is needed, if you see here, by increasing the carbon number, ethanol, propanol to butanol, so we are going from C3 to C4. The activation energy is increasing in both the cases. But the overall delta, the difference between the activation energy remains same. So this was one of the observations. Motivated from this, because we did the aviation fuel, we did the gasoline, we also moved to green diesel. And what we did, we changed the molecule, just this ethanol by butanol. And we were successfully able to identify that we can make the green diesel, and that all these molecules have been synthesized below 100 degrees Celsius. which is in contrast to the existing processes, which operates around 800 degrees Celsius or 700 degrees Celsius and uses hydrogen. Herein also what we found that the activity of the catalyst is directly correlated with the acidity as well as the kind of addenda atom and the central atoms we are having. Besides that, if my number of protons, like here you see 4 hydrogen, 5, 6, here 3, and then addenda atom is changing. So the catalyst activity without doing any experiment in actual can be calculated theoretically, and we can synthesize a wide range of new catalysts. And that all will be helpful. Either we make green diesel, we make green gasoline, or we make sustainable aviation fuel. So this is one part that we have done on the transportation fuel. Similarly, the another major emitter of the CO2 is the steel industry. So there what we are doing, we are converting this biomass into biocoal. And this biocoal has potential to straight away reduce the CO2 emissions by 40% in the steel industry. Similarly, there is one more thing that Dr. Joshi earlier highlighted during his plenary talk, that we are not talking about the methane, this part. So IIT ISM Dhanbad, we are in the coal mine region, and coal mines has a lot of coal bed methane. And coal bed methane, as of now, is left and it's a greenhouse gas emission. So it causes at least 25 times more CO2 emission. So what we are doing, we have taken this coal mine methane, I'll skip the data part, and we are converting this coal mine methane into the carbon nanotube and hydrogen. And carbon nanotube sells around 5 lakhs rupees per kg. So the technically or theoretically, I can say the hydrogen becomes free of cost. These are few of the carbon nanotubes that we have made. This is another example, because methane is the smallest molecule. We went for plastic also, which is the largest hydrocarbon molecule. And we are able to make here, I can show that we are able to make the hydrogen very successfully. So this is all. We are trying to align our research with the different initiative of the government of India and the vision of the Honorable Prime Minister. And these are my funding agencies. Of course, DBT has been very instrumental in initiating this transportation fuel and aviation fuel research. And this is my group. We got last year awarded also by the Petroleum Minister for CO2 emission reduction. And this is our HCBM car. These are my collaborators. Thank you. Thank you so much.
Thank you so much, sir. This is good, and we can think of how can connect the intermediates of SAF, and this is one of the burning issue in recent times, and current situations always lead to innovate toward and scale up towards production of sustainable aviation fuels or any kind of intermediates as well as other biofuels. So obviously this is very futuristic approach, and world is looking towards the future fuels everywhere. So whatever towards future fuels we can contribute, we have to contribute. The thing is how we can integrate this approach with the other leads, those has been generated till butanol or bio-based butanol, we can say. So that we can think of further. Thank you so much for enlightening us.
Thank you, sir. Thank you so much. Thank you.
So now we are going to serial number 7. Here the speaker is Professor Puneslo K. Badri for Indian Institute of Science, Education and Research, Kolkata. So this is how can microbiomes drive sustainability and bioeconomy, learnings from the field. So it's over to you, sir. Then we will take the presentation of Dr. Meghna. Please go ahead, sir.
Thank you, sir. Can you see my screen? Is it visible?
No, sir. No. You have to share the entire screen. Just go to share, then share entire screen and open the slides.
Okay, sir.
You had not shared yet anything.
Okay, sir. Let's go. Now, sir?
Perfect, sir. Perfect. Please go ahead.
Thank you very much, sir. Thank you, Dr. Kasturi ma'am and Dr. Balendra Singh for this opportunity. I'll very quickly show how the indigenous microbes can be used to address critical questions linked to sustainability and also drive the bioeconomy of our country. The first example I'll give you is here. Here is an example where we have done community microbiome engineering, and top left hand side you can see we have deployed these community microbes without any genetic engineering, of course, put them in the inlet systems of two big sewage treatment plants. One is 8.3 mld and one is 6.5 mld in West Bengal. And what we have shown is that these intelligent microbes, they can remove the fecal coliform in 30 days. Essentially, there is continuous flow of it, and the fecal coliforms are getting removed, the organic pollutants are in two major landfill sites, which produces huge amount of methane in Kolkata and in Guwahati. And what we have able to show is, top right hand side, if you look at this graph, between 50 to 100% removal of the methane emission post the application of the injection of these intelligent microbes, and all of these are indigenous microbes. So essentially, this system holds for about 6 to 10 months, so we can actually volume wise remove the methane emission, and then it can be then linked to this the recovery of the precious metals or any one of the other RED and other stuff. And so essentially the landfill sites can become safe, and of course the release of this leachate, that will be much safer. And now all these learnings has helped us to use again bunch of microbes which are of course indigenous. We have integrated into nano composites, and here is one example where we have bioremediated completely 1 kilometer stretch of a polluted river in Assam. And now the river is actually functioning. I do not know whether you see or not that, but the bottom right hand side image of April 2024, the fish population is back, and this is connected. This river is connected to Deepor Bill of Assam, which is an Ramsar wetland of international importance. So it's also sustaining the livelihood of the communities without any other additional intervention. The microbes are driving the what we call it as the passive restoration of the rivers. The top bottom right hand side image is another interesting work that we are doing, where we have put the microbes to in one of the biggest wetlands, 40 hectares. It has received for 20 years, leather effluent, and is under the Amrut mission of the government of India. We are slowly restoring this wetland. And what has happened by now is since the intervention, the Putias population, which is a very sensitive fish to pollution, it has returned back. So essentially showing that this can work. Very quickly, the bottom right hand side image is another example where we have used these microbes in Leh and Ladakh sector also to tackle water quality on a large scale. I will now shift, because of lack of time, that this knowledge of microbes are not only restricted to urban areas. We have now used these microbes in a combination with indigenous oyster population to slow down coastal erosion in mangroves, particularly from Sundarbans. Professor Krishna Roy talked about it, and it has shown remarkable result where we have more or less managed to hold the islands where there was high rate of erosion using the combination of these microbes and the oyster population. Last but not the least, I want to give an example very quickly where we have used the microbes in shrimp ponds, and we have brought in this concept of mangroves with shrimps. Essentially, the farmers are no more using antibiotics, no more pesticides, nothing like that. The shrimps that are growing are completely healthy, organic, and now they're getting exported to Europe and Japan. Again, using microbiome learnings from the field. And I'm very pleased to share that this has received the Global Technical Award of 2025 from the Food and Agriculture Organization of the United Nations. I come and end the last slide, which is the bioeconomy driven slide, of course, with the connecting the previous one. And here is one example where the indigenous cyanobacterial population has been used, scaling up has been done up to 1,000 liters to produce dolastatins. Dolastatin is a very important drug scaffold for tackling cancer, So we are able to do that, and we show that could be a very sustainable method for actually producing bulk volume of dolastatins, which is an immediate need for the pharma industries, and they're desperately looking for it under the Atmanirbhar Bharat scheme. What I want to highlight is that DBT has very generously supported us. Our learnings from the microbes and the nanocomposites, now we are able to show that it can remove 50% of the common microplastic pollutants, particularly PET and polyethylene, under control conditions, and we hope that in future we'll be able to use these learnings to apply in the field under control condition. We are very thankful for DBT to supporting us, and also our lab members who have done all the work. Thank you, sir, for this opportunity.
Thank you very much, sir. Thank you very much to take all of us and all the participants through one of the universe of different activities, and it's fantastic to connect how microbes can be used, or how these microbial cultivation, algal cultivation methods can use and can contribute towards the larger large scale global problems. So really, this can be helpful in addressing environmental issues and climate change issues. So thank you so much for connecting us with this all. So now we are moving to number 10. This is dry times at forest stages, seedling traits in response to Dr. Meghna Krishnadas from NCBS Bangalore. Can you hear us?
Good afternoon. Yes, I can hear you. Can you hear me?
Please go ahead. Yes, we can hear you. Please go ahead.
Okay, let me just share my screen. Yes. So just give me 1 second. Can you see my screen?
Not yet, ma'am. Kindly go to share button and bottom.
Yes. Panel. Can you see it now?
Yes, perfect.
Oh,
please go ahead.
Okay, thank you so much. Well, first of all, thank you so much for this opportunity to present my ongoing DBT project, which is to understand how forest regeneration varies in response to potential changes in the climate and in human-modified fragmented forests of the Western Ghats. Thank you so much to DBT for funding this work, and we are pleased to see that a lot of the work that has gone on is already showing successful results. So to start off, just to set the context, in today's world, human activities have altered ecological processes and ecosystems at the planetary scale. And these massive global environmental changes are having effects on different species and ecological communities. And a major scientific goal is to understand how species are going to respond to these changing times. Now, while individual threats such as habitat loss or climate change have seen a lot of work, what still remains understudied is the interaction between these forces of change that ecological communities do, which is to understand that if you have climate change, which causes issues like drought in the future, how will that react with another problems such as habitat fragmentation, which creates these edges that disrupt what ecological communities look like. So let me just explain this to you. When you have a large patch of forest, any patch of forest will have some area that is comprises of the edges. But when you break up a large patch into smaller fragments, you increase the proportion of edges in that landscape. Now, edges tend to be warmer and drier, they're subject to desiccation. So logically, we would think that with climate change, with there being a drought, edges of the forest are going to respond or be affected differently from the interiors. We can also think of this in a way that species that are, that can colonize at edges will now be restricted not only by the local changes, but by the drought effects that might happen, and forest interiors might serve as refugia where some kind of species can persist only there. So we wanted to understand some of these from the characteristics of their leaves, their stem, their roots, their physiology, especially for plants, which determine how they use resources and respond to different stressors like drought or other changes that might happen. And we wanted to do this in an experimental framework by setting up a potential drought situation in the field itself. So this was an ambitious experiment that we were trying to do. We went to a forest. We've been working here for almost a decade now, so we know the landscape very well. And we set up these experiments, if you can see my cursor, where we had these paired sites at the forest edge and the interior, where we transplanted seedlings of 16 different species that we grew over a period of 1 year from seed into seedling in controlled greenhouse conditions. And then one of these treatments, we imposed a drought shelter, a rainout shelter, which reduced the availability of soil moisture. And because we've done pilot studies before, we know that soil moisture decreases considerably with these drought things. And even in this study, we see that the response of soil moisture becomes most prominent in the dry times, which supports one of our hypotheses that forest edges, when things get drier, edges are likely to get more dry than interior forests. Now, first of all, when we measure all of these traits among these species, we find, interestingly, that the water use traits, which is traits related to stomatal anatomy and turgor loss point, which is a key physiological trait, and other anatomical traits related to water use, they form a completely different dimension of species strategies as opposed to the morphological traits. And so far, most studies have only focused on morphological traits. So we found that this understanding water use traits is seemingly really critical for tropical forest seedlings. And in that, mostly we need to look at this one trait, which is called turgor loss point, which is basically the water potential at which species start wilting, the leaves start wilting, a key indicator of their drought tolerance. The more negative this trait is, the more tolerant a species is of drought. So we have characterized this, and we see that even among what are called wet, humid forest species, there is substantial variation, which is a very important insight that we have gained, which means that species will respond to drought differently based on their turgor loss point. And by the way, this is all data still being collected. So what I'm presenting here is quite preliminary. And in a future presentation, I hope to have more robust results. But even in these preliminary data, what we see is that species hydraulic safety margin, which is basically the difference between the minimum water potential that they experience in the soils that they're in comparison to their turgor loss point. This is a great indicator of how well they are doing in drought. The lower it is, or the closer it is to zero, or if it is negative, it means they are under drought stress. And very clearly, you see that regardless of whether they are at the edge or in the interior, the drought treatment that we have imposed pushes them closer to their safety margins, which means that they are experiencing drought stress because of a loss of soil moisture. But this differs among different species, as you would expect, because they differ in their physiological tolerance to water scarcity. But most importantly, what we find is that the closer they are getting to their safety margin, it's actually more intense when you have drought in the forest edges, which is exactly what we hypothesize, that edges will make species more susceptible to drought, which means we need to take these steps to conserve species at the edges because they are being more susceptible to drought. We see this in species responses in terms of their mortality as well. There are some species like Holigarna nigra and Symplochos racemosa, which are showing much higher mortality in drought compared to controlled conditions and more prominently at the forest edges. But of course, there are some other species where you don't see that much of a difference, which might be related to their hydraulic safety margins or their turgor loss point, as I mentioned. So these were preliminary results. The next steps are that we want to have these more robust statistical models to understand species drought responses. And these responses will be in terms of their survival, as I just presented some data here, their physiological responses in terms of how much they're photosynthesized and the growth of their biomass. We also want to understand whether it's individual traits or the whole plant phenotype that explains drought response. and how whether this explains the difference between edges and interior. And why is all this important? Because understanding how species might respond to drought and microhabitat conditions is going to help us plan better for restoration by choosing appropriate species. And the next step, of course, is using all of this data to make some predictive models of what the future seedling community is going to look like, for which we have already initiated some collaborations with modelers, and something that my lab also does. And with that, I would like to acknowledge my student Gayatri, who has been leading this project in the field and doing a fantastic job. But along with this field team, which I'm very grateful to, there's a lot of hard work collecting seeds, growing seedlings. It takes a year before we even start the experiment, and it's a lot of hard manual labor. And these people have done an excellent job, so I thank them for it. Of course, Department of Biotechnology for funding this project, and TIFR and CPS for the support that they've given in terms of logistics and others. With that, I'm happy to take questions. Thank you so much.
Yes, thank you so much, ma'am, for sharing the fantastic insights and how you are working to define the traits on the edges or forest edges, and how it can be important to establish further the predictive models based on this. Really, once it is established for one, definitely it will work for other traits also, and it will lead to somewhere to some important solutions from climate angle. And really, water stress is one of the major issues that is happening because of the climate change. And this has to be addressed in our forest, and forests are obviously very much important to mitigate climate change further, or towards consolidating our mitigating efforts. So forests are almost on edge of the climate, we can say. So those edges
Yes.
has to be defined and addressed. Thank you so much, ma'am.
Yes. Thank you so much.
So
Thank you.
now,
Yeah.
so now we are at serial number 8. This is pollinators and ecosystem services in the Anthropocene. And the speaker here is Dr. Hema Somanathan from Indian Institute of Science, Education and Research, Thiruvananthapuram. Thank you so much, ma'am. It's over to you, ma'am. Please finish in 5 minutes.
Can you see my screen? Hello?
We can't see yet. You had not shared yet, ma'am.
Okay, let me try once again.
Just go to share button in bottom panel.
That's what I did. Share. Entire screen is what I selected. Can you?
Not yet shared. Oh, after selecting entire screen, then again you have to press the share button.
Yeah, the share button is not active over here. Only the cancel button is active.
I think some pop active in your. Yes, yes, yes.
So that is breaking. There is some connectivity issue, I guess.
We cannot hear you, ma'am, actually. Acha.
Is a western honeybee, but that's an exotic species.
Please go ahead.
Ma'am, I I think we can't see your screen and you are not audible. You need to unmute.
Can you hear me? Hello?
Ma'am, can you hear us?
I can hear you clearly. Yeah.
Yeah, your share screen has stopped. You need to reshare the screen.
invasive fishes, which can cause major havoc in the Ramsar wetlands. Thank you, and this is just the first year of the project, and we are hope to continue to do this across other Ramsar sites. We'd like to thank the Department of Biotechnology for giving this opportunity and also for funding this particular program, and Dr. Robin Raja, who is leading this project as part of this study. Thank you very much.
Thank you so much, sir, for sharing these insights on alien fishes and native biodiversity threats. Obviously, ATRI is the very important stakeholder for all the biodiversity-related problems, and biodiversity conservation is very much linked in both ways, either for threats due to climate change or for action towards the climate protection. So that biodiversity conservation or management, we can say that is very much important. And ATRI is contributing very much towards this cause. So thank you so much for sharing the insights. So, ma'am, we are done with the technical program. We can take some QA. If there are some questions from online participants, 1-2 questions only. So there is one question from online, from YouTube to Dr. Azaj Ahmed. The question is, can biobutyric acid and biobutanol can be considered to use in SAF production? Dr. Azaj Ahmed, sir, it's for you. I think he's on mute or...
Pass on these questions to respective speakers.
He has gone out for a minute. He will be here in a minute, ma'am. Professor Ijaz Ahmed will be here in a minute, ma'am.
We can wait for 1 minute. Is there any other question from any of the participants?
Yeah, sorry. So I went for water.
Be used for SAF production in coming days. So this is from participants from YouTube. Biobutyric acid and biobutanol can be considered to use in SAF production.
Yes, so exactly this is what we are doing. So when butanol is produced, butyric acid comes as a byproduct. %50% for blending.
Great. So one more question is there. This is very general question. So he says there are various efforts being made under research and development for biodiversity conservation and forest protection, as well as other conservation initiatives. So why the achievements made in the laboratories are not coming to the in kind of awareness in general public? So that is kind of concern. So can somebody comment on this?
So I would say this is a very good question. If I answer from my perspective, the real challenge is when you develop a technology in the lab. After that, it requires a lot of people from mechanical, electrical, electronics as a EPC consultant to scale up. And right now, that link is missing. So we develop the technology, but for a scale up, we need a bigger team to come together.
Any other question? If not, there is one suggestion from somebody on YouTube. So they say, let's promise to plant more, waste less, and love the earth. So with this, the program is over to you, ma'am, for your closing remarks.
I would congratulate all of you for the excellent research and achievements what you are continuing, and would wish you all the best for future. And what is important that we need to translate this research. So in the field, and most of you are doing, so there are some few examples which I have seen, like mangroves and all, those are happening in the field and which is really creating the impact, and that has been demonstrated. So request all of you, all the project investigators, once again, thank you all of you for joining and presenting your work in very short time and short notice, and with enthusiasm. I think he has written the poem. That is also a compliment to Dr. Valender for the, he has been writing poems on this, and we all are, together we can create real impact towards the sustainable development and how to protect our environment. With this, I would thank all of you once again, and over to you, Balin.
Thank you so much, ma'am, for giving this opportunity and inviting all and providing this platform to share the thoughts and developments happening in the different programs under your esteemed guidance in forest biotechnology or advanced biofuels under the Ages of Biomanufacturing program. I also thank special thanks goes to today's Chief Padma Awardee, 2 times Padma Awardee, Padma Shri, Padma Bhushan, and one of the great environmentalists for telling us about the impact of the smaller initiatives and how they can lead to big changes from climate point of view, and how our small efforts at individual levels like carbon footprint mapping and this can help in building the resilient ecosystem. And he has delivered it in the manner that one common man can understand. That's very important. I thank you on behalf of this program division and Department of Biotechnology. The special thanks also goes to Secretary Department of Biotechnology for allowing us and guiding us with its leading remarks how the further activities can under biomanufacturing can provide and enlighten us to work further for development and expansion of different programs on the lines of national priorities under climate change. I thank each of the investigator very much for coming on this platform on very short notice and sharing the insights. I cannot explain each of that, but we have received very good comments. I mean, very good comments on the YouTube and as well as from online participants. Each of your lecture and each of your slide, whatever you had delivered here, and many of the students had commented there. So thank you very much for sharing these insights. These will help, somewhere transforming the programs being built by Department of Biotechnology. Thank you so much. We will work together. Virtual link. Thank you for MOFCC. They have delivered the converging policy remarks. How the government of India is making the wholesome efforts towards climate change mitigation. So thank you all of you again. Thank you very much for all participants, attendees, students, teachers, faculties, everyone. Who has I cannot name everyone, but everyone. Thanks goes to everyone. Thank you very much.